everyone. We are now in our second lecture and we're going to be looking at cells. Okay, so we're going to be looking at the structure of cells, all the different organelles in those cells. And then after that, we're going to go into what's called the cell cycle, which is basically just the life cycle of a cell. We're going to talk most of our time on mitosis, which is cell division, making more cells from a cell. Okay, so when you look at a cell, usually in a textbook, they give you a picture similar to this. This is what we call like a generic or a typical cell. I'd like to point out right off the bat that a lot of cells don't look like this. There's a lot of specialization out there. But when you look at a generic cell, this is generally what it looks like. Now, we talked about cells just a little bit last time. Do you remember when we defined a cell? In textbooks, they often give you that a cell is the basic unit of life. So let's look at that a little bit further, make sure we really understand what that means. So unit, when I think of unit as a piece of a whole, right? So you have a piece of a whole, and then basic usually in this case refers to the smallest. So what we're looking at is that the cell is the smallest piece of an organism that's considered to be alive. Now remember, there were smaller pieces. You have atoms and molecules and macromolecules and organelles. Those are smaller, but they are not considered to be alive. They do not reproduce. They do not respond to the external environment. They are not alive. Okay, so the cell is the smallest piece of the whole that is considered to be alive. Okay, so our bodies are made from cells. Okay, so now let's get into the pieces and the structures of the cell, starting with the outside, which is called the plasma membrane. Another term for plasma membrane is cell membrane, which it's just the outside of the cell. You should definitely be familiar with the term plasma membrane though, because that's what's on your lab list. Okay, so the plasma membrane is just the outside membrane that goes around the exterior of the cell. And the reason you have a plasma membrane is so that you can have an external boundary. So that's really the major function of that plasma membrane, so that you can make sure that a cell is distinct and separate from everything on the outside, right? You can't have like a living cell if it's the same as everything else. Right, so the purpose of that plasma membrane is that it allows the cell to decide what to bring into the cell and what not to bring in the cell, and that creates an internal environment that's separate and different from the external environment. So basically, you got a sack of water with different water inside than outside, different stuff dissolved in it. That's basically what it boils down to. Okay, so the plasma membrane is what we call selectively permeable. The word permeable means able to pass through, and so selective means some things can get through and other things cannot, and that's how it's able to create an environment that's different. It's selectively permeable, or sometimes referred to as semi-permeable if you see that term. Okay, so let's look at the structure of the plasma membrane. It's what we call a phospholipid bilayer. Kind of a scary word, so let's break it down. Okay, first of all, when you see bi, bi means two. So we're going to be looking for two of something. And what we're looking for is two layers. Okay, so the majority of the plasma membrane is made up of a molecule, a macromolecule actually, that is called a phospholipid. And the phospholipids are special structures because they have two regions to them. They have what's called a hydrophilic region and a hydrophobic region. Hydrophilic, hydro means water, philic means loving, that's water loving. Hydro means water, phobic means fearing, water fearing. So I actually don't want to get too far down the path of the chemical properties of the phospholipid bilayer. If you go on to physiology, you'll get into that quite a lot. But for now, I just want you to understand that there are two layers of phospholipids, and their tails, which are what you see here, are all facing each other in the middle, and the heads are on the outside, and they create two layers. And this is what makes it very, very good at being a selective membrane, because a lot of things that are hydrophilic, by the way, a lot of things are unable to pass through the phospholipid bilayer. Okay, so again, a lot of information. Just want you to understand phospholipid bilayer is two layers of phospholipids where the tails are in the middle and the heads are on the outside. Okay, so most of the plasma membrane is made up of those phospholipids, but embedded in that phospholipid, you have a bunch of other things. Most of those other things are proteins. Okay, so they create what's called the fluid mosaic model. And what they mean by that is, think of it this way. So if you think about the ocean, right? On the ocean, the phospholipids create this kind of watery ocean. So think of the water as the phospholipids. And then floating in those phospholipids, that's where you're going to have your embedded proteins. And those are things like receptors that bind to external molecules, or things like enzymes, which are proteins that have a particular function. They do chemical reactions. Things like identity markers that say, hey, I'm part of the cell. Okay, so they just sort of float around within that phospholipid bilayer. Okay, so again, the majority of the plasma membrane is made up by these little phospholipids that create the phospholipid bilayer, two layers. Okay, so let's look at some appendages of the plasma membrane. And when I say appendage, I just mean things that stick out. That's all. That's what I mean. Things that stick out. So if you think of like appendages for our own bodies, those are like arms and legs. So the appendages for the cells, those would be things like cilia, microvilli, and flagella. So let's look at what those look like. In this picture here is a cell right here. And if you look at this picture here, you'll notice that at the surface of the cells, you have a bunch of really tiny little hairs. Those are cilia. So all of these structures right here, those are cilia, and those are just little hair-like extensions that come off of the surface of the cell. And what's really important for future, and we'll look at it later, is that the cilia move. Okay, and so not only do they just stick out, but they also move together in a coordinated fashion. And when they're all moving together in coordinated fashion, they can create currents for the liquid that surrounds the cells, and those currents can move things around in the body. So for example, in the uterine tubes, okay, so bringing the egg from the ovaries into the uterus, you have a bunch of those cilia. And so the way that the egg actually travels from those ovaries all the way to the uterus is because the cilia beat together in a coordinated fashion, and that makes the current that sweeps the egg along towards the uterus. Okay, so cilia move. And the reason I highlight this point is that there is another type of appendage called a microvilli. And a microvilli honestly looks very, very similar, very, very similar to a cilia. Both of them are little tiny extensions that come out of the surface of the cell. Okay, the big difference between cilia and microvilli is that cilia move. They move things along. Microvilli do not move, okay? No sweeping, no movement, none of that. Their job is simply to increase the amount of surface area. And for those of you who are unfamiliar with what surface area means, it basically just means the amount of space on this outside of an object, okay? So if you think about like a box, like you're wrapping a box, you have the volume, which is the internal space, and then you have the surface area, which would be the amount of space seen on the outside. So one of my students once told me, surface area is like how much wrapping paper will you need to cover the outside of that box? That's surface area. And so surface area will become very important anytime you're moving things across into or out of the cell. It'll come back to us when we talk about the digestive system, absorbing nutrients into the body. It'll also come into play when we talk about the respiratory system, how you get oxygen and carbon dioxide into and out of your lungs. Okay, so surface area is going to become important for that. And one of the ways that your body is able to increase the surface area is by having these little projections. Okay, so you have these little projections upwards, and that will help increase the amount of external space, which makes it easier for things to pass into or out of the cell. Okay, another slight difference is that microvilli tend to be shorter than cilia. Cilia tend to be longer and more elaborate, but honestly, it's very difficult to really tell based on that. A better way to just a description of how they work. Okay, so how are they different? Which one moves? Cilia moves, it sweeps things along, and microvilli do not move, they just increase surface area for increased absorption or secretion. Okay. Okay, last little appendage coming off of the cell, plasma membrane is the flagella. And I'm not going to spend too much time about it. And the reason for that is that there's only one cell in the whole body, of the whole human body, not all of us, in fact, that has a flagella. A flagella is a very long, extended tail-like structure. It's used for locomotion. And what locomotion means is moving the whole cell along, so swimming. So what cell in our body has a long tail and is used for swimming? The answer is a sperm. Okay, so we probably won't see flagella too often. We will revisit this when we get to the reproductive system. We'll look at the structure of sperm more closely, but for now, make sure you definitely understand cilia and microvilli. Okay, so let's go into cytoplasm. Cytoplasm is just the fancy way of saying everything between the cell membrane, the plasma membrane on the outside, and the nucleus in the inside. Inside of that cytoplasm, you have a bunch of fluid. The fluid itself is called cytosol. So basically, that's just water with a bunch of stuff dissolved into it. And then floating inside of that is the organelles. Organelles are particular structures that do a particular function for the cell. Okay, membrane-bound structures that do something for the cell. Okay, so everything in there, from nucleus all the way to the cytoplasm, so everything that's light green right here, including all of these organelles that are shown here, all of that collectively would be called cytoplasm. And the other thing that I want to talk about is the cytoskeleton. Now, let's take a minute because I forgot to mention it in the last slide. Let's look at this prefix cyto. That is going to come up a lot. Okay, so for those of you who are doing your running list, which I hope is all of you, on your notes, the very first front of your notebook, your running list, go ahead and add the prefix cyt, C-Y-T. Usually it has an E or an O. Okay, that is going to mean cell. Okay, so cell. Okay, so we saw here already cytoplasm. That's the liquid, plasm liquid, cell, and then cytosol, which is the liquid inside of it. Okay, so let's look at a cytoskeleton. Inside of the cell, oh, I should move this picture. Inside of the cell, you have long protein filaments. And what I mean by that is you have a whole bunch of individual proteins that hook together. And when they hook together and they coil around each other, they create long, long strings of protein. That's a protein filament. Now there's several different sizes and several different types of protein filaments. You've got microfilaments, micro means small, remember, intermediate filaments, medium size, and then microtubules. But collectively, when you look at all of them, those microfilaments, those protein filaments inside of the cell, they create a framework for the cell. Okay, so I like to think of it sort of like the scaffolding of a building, right? If you're building a skyscraper, you put your scaffolding up first and then you build off of that. So a major function of the cytoskeleton is to provide structure for the cell. Okay, so let's look right quick at why it's called cytoskeleton. What does the skeleton in our own bodies, our skeletal system, our bones, what do they do for us? First of all, they provide shape and structure. Most of the shapes on your face, for example, come from the bones that are underneath them. They also provide support against gravity and allow for, and the last thing that we almost always forget is that your skeleton is important for movement, because yes, your muscles contract, but they wouldn't do anything if they didn't have anything to pull on. They need a lever to pull on. Okay, so that is what the cytoskeleton does for the cell. It provides shape and structure. If you look at this picture right here, this is a histology where we're looking at the cell, and what we're looking at is all of these microtubules where they've been, we have a bunch of fluorescent proteins connected to them so that they shine green in the microscope. That's what we're looking at right here. Okay, so green fluorescent proteins on microtubules. And you can see that those microtubules, they come together to create the scaffolding, which creates the shape and the structure of the cell. Okay, now the other one is a movement. So this is going to become especially kind of obvious when we get to the skeletal muscles, right? Skeletal muscles, they have to do a lot of movement. And that movement would not be able to happen unless you had something to pull on. Okay, so the cytoskeleton allows for movement, that things like moving the cilia around, or the flagella, but also things like contraction in the skeletal muscle. Okay, so support, structure, and movement. That's the cytoskeleton. Okay, so I'm going to go ahead and stop this maybe now. I'll go on and do another piece on organelles and the nucleus, and we'll talk about not just what they are, but their functions as well.